Most animals are born to die. But in the farthest corners of our planet, a handful of species have rewritten that rulebook. From a shark whose eye proteins have lasted since before the Titanic sank, to a jellyfish that can literally reverse its biological clock. These life forms force scientists to ask, are the rules of death negotiable? If humans are prisoners to aging, how do these animals escape its grip? What secrets do their cells and chemistry reveal? And what does that mean for us? The evidence begins with a specimen that turned back the clock on everything we believed about lifespan. Aboard the trawler Helga Maria, the deck pitched under gale force winds as researchers scrambled to secure their catch. Among the tangled nets, a Greenland shark stretched nearly 5 meters, its mottled skin hinting at an age few could guess. Biologist Julius Nielsen reached for the most valuable sample, the shark's eye. Not the retina, but the lens, the hard, crystal-clear core that forms in the earliest months of life and remains unchanged, sealed off from the rest of the body. This eye lens, isolated with gloved hands and stored in a sterile vial, became the key to unlocking the animal's true age. Back in the lab, the lens core underwent a process more familiar to archaeologists than to marine biologists. Technicians measured the ratio of carbon-14, a radioactive isotope boosted by Cold War nuclear tests. The bomb pulse curve, a spike in atmospheric carbon from mid-century detonations, created a timestamp in living tissue. But for this shark, the lens core showed a carbon signature from long before 1950, untouched by the atomic age. Each year, as new layers built around the original nucleus, only the outermost proteins reflected the world's changing chemistry. Deep within, the oldest proteins remained as they were at birth. The data pointed to a birth year somewhere in the early 1600s. This specimen had been alive as Galileo peered through his telescope and Rembrandt painted his first canvases. The shark's eye lens, preserved through centuries of Arctic darkness, provided the most reliable biological clock ever found in a vertebrate. No other method, no tooth and no bone, could match the precision of this forensic breakthrough. The result forced a rethinking of what it means to age in the deep sea and set the stage for a new question. What inside these animals keeps their vital machinery running for centuries when nearly all other life succumbs to time? Inside the Greenland shark's eye, a molecular puzzle unfolds. The lens is built from crystalline proteins, folded and locked into place during the shark's first months of life. Unlike most animal tissues that constantly renew and repair themselves, these proteins are set for the duration, unchanged as centuries pass. In laboratory assays, shark lens crystallines resist unfolding even at temperatures just above freezing, around -1 degree Celsius. They keep their shape, their clarity, and their function, while the world outside the shark changes from wooden ships to nuclear submarines. This remarkable stability is not just a quirk of the eye. It hints at a deeper biological principle, one where certain tissues are shielded from the usual wear and tear that leads to aging. Crystallines act as molecular chaperones, preventing aggregation and clouding, even as decades pile up. In contrast, most vertebrate proteins slowly unravel or clump together, clouding vision and signaling the decline of old age. But in the Greenland shark, these lens proteins maintain their structure far beyond the lifespan of any other known vertebrate. Researchers have begun to wonder if this selective resilience extends beyond the eye. Are there other organs where time seems to pause, where proteins and enzymes remain as robust at age 200 as they were at age 20? Early findings suggest that the shark's metabolic machinery, especially in cold-adapted enzymes, shows a similar resistance to breakdown. Unlike human tissues, which accumulate damage from reactive oxygen species, and metabolic byproducts. The shark's slow metabolism and cold environment may help preserve its most vital molecules. Yet organ-specific maintenance raises new questions. If the eye can remain ageless, what about the heart, the liver, or the brain? Is the shark's longevity the result of a few privileged tissues, or does its entire body share in this molecular resilience? The answers lie deeper, in the interplay between environment, genetics, and the quiet, relentless stability of proteins that refuse to yield to time. Cells in most animals are locked into their identities. Skin stays skin, nerve stays nerve. But in Turritopsis dohrnii, the so-called immortal jellyfish, the blueprint is far more flexible. Under stress, an adult medusa can collapse into a gelatinous cyst, then reawaken as a juvenile polyp, as if a butterfly could shrink back into a caterpillar. The secret lies in transdifferentiation, a cellular process that rewrites fate itself. Inside the cyst, a wave of genetic activity sweeps across the genome. Chromatin, the tightly packed material that controls gene access, begins to loosen, exposing regions needed for repair and development. Genes associated with DNA repair surge to the forefront, some of them unique to this jellyfish. Nearly half of the most active genes during this transition have never been seen in any other animal. Their functions still a mystery. The rest are familiar. Enzymes that patch up broken DNA, factors that silence aging pathways, and regulators that unlock pluripotency, the ability to become any cell type. This is not simple regeneration. The process involves a wholesale reprogramming of cell identity, orchestrated by signals that resemble those found in early embryos. While most animals rely on dedicated stem cells to repair damage, Turritopsis cells can leap directly from one fate to another. The result is a biological reset, erasing the scars of age and injury. For scientists, the question is whether these mechanisms, chromatin remodeling, repair gene upregulation, and pluripotency activation, could one day inform new strategies for human medicine, or if they remain a peculiar trick of jellyfish evolution. Either way, the rules of cellular identity are not as fixed as once believed. Shin Kubota's lab was little more than a cramped room above Kyoto's saltwater canals, but for over a decade it became the world's headquarters for a single, stubborn experiment. Each morning, Kubota logged the temperature and salinity of his tanks, then peered through a microscope at hundreds of Turritopsis dohrnii jellyfish, each one a possible escape artist from death. The early attempts were failures. Most jellyfish collapsed under stress or succumbed to bacterial infections, echoing the high mortality rates that plagued aquariums since the 1920s. Kubota refused to give up. He sterilized every instrument, filtered the water, and worked late into the night, recording every setback in his notebooks. The breakthrough came not from a new technology, but from a change in diet. By supplementing the jellyfish's meals with arginine, a simple amino acid, he tripled the survival rate of his colonies. The polyps became more robust, and for the first time, Kubota could reliably induce the life cycle reversal. Under controlled stress, adult medusae shrank into cysts, then sprouted back into juvenile polyps, repeating the cycle again and again. Over 10 years, he documented each successful reversion, building a diary of what practical immortality looks like in a petri dish. The process was never automatic. It demanded constant vigilance, axenic maintenance to prevent contamination, careful management of stress triggers, and a strict feeding schedule. Funding ran thin, and Kubota's handwritten logs sometimes recorded more hours than meals. Yet his persistence paid off. The world's first stable, self-renewing Turritopsis colony proved that the rules of aging could be bent, at least under the right conditions. The price of immortality, it seemed, was relentless attention and a willingness to rewrite the protocols of marine biology, one entry at a time. On the seafloor beneath the Pacific Northwest, glass sponges build their homes from the simplest material, dissolved silica. Over centuries, they draw minerals from the water, assembling intricate lattices that rival the strength of steel yet remain almost weightless. Each sponge grows into a living greenhouse, its skeleton a web of glass needles that traps water in a maze of channels. Here, the rules of survival are pared down to their barest form. No gut, no moving parts, only passive filtration. Seawater drifts through the sponge's body, carrying microscopic food and oxygen. As long as the water stays clear, these ancient structures endure, some reaching ages of 11,000 years. But endurance comes with a price. Even a thin cloud of sediment can bring a sponge's life to a halt. When dredging or mining stirs up the seafloor, particles settle onto the sponge's delicate lattice, clogging its pores. Research from 2023 shows that a heavy sediment plume can stop a sponge from feeding in less than 2 hours. Without flow, the cells suffocate, and the body collapses. A single mining event can erase centuries of growth in a single afternoon. For the Haida people of Haida Gwaii, these sponges are more than biology. They are timekeepers, guardians of ancestral memory. An elder once said, "Sponges hold our ancestors' stories. Miners erase them. The lesson is written in glass. What endures in the deep depends not on complexity, but on a fragile balance between resilience and vulnerability." I am Dr. Maya Quinn, an ocean biologist, and this is a closer look at black coral, one of nature's living time capsules. A black coral colony grows so slowly, it is measured in millimeters per year. Some of the oldest specimens, hidden in the depths off Hawaii and in the Mediterranean, have been alive for more than 4,000 years. Each branch is a living record, storing chemical traces of seawater chemistry from centuries past. That record archives volcanic eruptions, industrial pollution, and the rise and fall of civilizations. These colonies are literal records of time. In 2022, a planned submersible dive off the coast of Hawaii was denied at the last minute. Official logs cited the risk of disturbing the reef, a concern sharpened by the fragile, slow-growing nature of black coral. Scientists expressed frustration in private. Every year lost is another year of missing data, another gap in understanding how these colonies weather the planet's changes. The fragile reality of slow growth means a single disturbance can erase centuries of history. Permits for sampling are tightly controlled, and with good reason. A single careless collection can remove centuries of growth from a colony. Yet mining companies have secured access to nearby sites, and they argue their data is proprietary and their impact minimal. That argument has created a persistent tension between conservation and exploitation. Regulators must weigh the value of knowledge against the need to protect what little remains. For scientists, the dilemma is acute. Without responsibly obtained samples, the secrets of black coral longevity remain locked away. The debate centers on how to pursue discovery while preventing irreversible loss. Biologically, extreme longevity in black coral appears tied to a strategy of extreme slowness. Low metabolic rates, slow cellular turnover, and life in stable deep-sea environments reduce cumulative damage and slow the pace of aging. That stability is also the weakness. When the environment changes, these organisms cannot quickly adapt or recover. In that sense, longevity is a trade-off, not a free gift. Immortality in nature often requires simplicity or a dependence on conditions that do not change quickly. So we are left with a difficult question. How do we balance the urgency of scientific inquiry with the ethical duty to preserve living archives that have endured since before the pyramids were built? And what does it mean for humans, whose lives are short in comparison, to study beings that can persist for 4,000 years? That question asks us to reflect on how we value time, knowledge, and the fragile complexity of life. Patterns begin to emerge when the rules of longevity are mapped across the animal kingdom. 4 core strategies stand out, each a different route to resist ordinary wear and tear. This is a biological playbook, not a list of curiosities. The first strategy is genome maintenance. In long-lived species such as the Greenland shark and the Galapagos tortoise, extra copies of DNA repair genes act like a constant patrol against mutations. These animals invest heavily in fixing broken strands and correcting errors before they can spiral into cancer or cellular decline. Their genomes behave more like a well-edited manuscript than a draft full of crossed-out lines. Genome maintenance buys time. Next is cellular plasticity. The immortal jellyfish shows how adult cells can revert to juvenile states through transdifferentiation. That flexibility lets the animal erase accumulated damage and sidestep the rigid developmental pathways that limit most multicellular life. The process is like a software reboot, returning the system to an earlier, less corrupted state. What can human stem cell research learn from this adaptability? Plasticity points to profound possibilities. Redox control forms the third pillar. In the deep sea, low metabolic rates and cold temperatures slow the production of reactive oxygen species, the chemical sparks that cause oxidative stress. Species such as glass sponges and ocean quahogs thrive in these stable conditions, where tissues avoid the chronic damage associated with a high-energy lifestyle. For them, longevity is less about fighting damage and more about never letting it start. Redox balance underlies quiet endurance. Finally, ecological context shapes the boundaries of survival. Black corals and some sponges occupy habitats with few predators and minimal disturbance, environments that reward patience over speed. With safety and stability, slow growth becomes a viable strategy, and organisms can persist for centuries or millennia. Ecology defines the space where extreme longevity is possible. Taken together, these strategies reveal a playbook that is both diverse and deeply interconnected. They expose the trade-offs behind exceptional lifespans, often simplicity, extreme specialization, or dependence on a stable environment are the price of apparent immortality. That blueprint contrasts sharply with the human pattern of aging. If black coral can live for 4,000 years, what does the finite lifespan of humans tell us about the values encoded in our biology and the kinds of lives we are built to lead? What lessons can biology offer, and which trade-offs do we choose to accept? Human cells live under a different set of constraints. Every day, the body's 30 trillion cells generate energy by burning oxygen, a process that keeps us warm and active. But it comes at a cost. This metabolic engine produces reactive oxygen species, tiny sparks that damage DNA, proteins, and membranes. Over time, the wear accumulates. Even the best repair crews in our cells cannot keep up forever, and the result is the slow march of aging. Galapagos giant tortoises face the same threats, and yet they have developed a remarkable defense. Their genomes carry multiple copies of the TP53 gene, a master regulator of DNA repair and cell death. In humans, a single copy of TP53 patrols the genome, triggering apoptosis, programmed cell suicide, when damage becomes too great. Tortoises with up to 12 copies can detect and destroy damaged cells much more efficiently, stopping cancer in its tracks before it can take hold. Laboratory tests show tortoise cells self-destruct at the first sign of trouble, while human cells often let errors slip by, risking mutations that accumulate with age. The difference runs deeper than genetics. As ectotherms, tortoises rely on the environment for heat. Their slow, cold metabolism produces far fewer reactive oxygen species than the furnace-like cells of mammals. The result is a double advantage: less molecular damage in the first place, and a vastly improved system for cleaning up what little harm occurs. Humans, by contrast, pay a price for our high-energy lives. Our brains and muscles demand constant fuel, and the resulting oxidative stress is the tax we pay for speed, agility, and warmth. The question lingers: could boosting our own DNA repair pathways or dialing down our metabolic rate extend human life in the way it does for tortoises? Or are we bound by trade-offs between vitality and longevity, between complexity and resilience, that cannot be engineered away? The answers remain open, shaped by the same evolutionary forces that made us and the animals that seem to play by different rules. Biological immortality is not just a curiosity, it is a window into the mechanics of life itself. As researchers race to decode DNA repair, metabolic stasis, and cellular renewal, the secrets of these animals could reshape how we understand aging, disease, and even our own mortality. In a world chasing longevity, their existence asks a hard question: is living forever worth what we might have to give up? Share your thoughts below.